Can you hear me? Good, okay. It's my great pleasure to introduce Dr. Laurie West. She's we are thankful for her to come here and talk. And she's a well-renowned cardiologist and is a professor at the University of Alberta in the pediatrics. And she also has appointment in medical microbiology, immunology, Department of Pathology, etc. And she's also director of Alberta Transplant Institute. And she's also the director of Canadian Donation Transplant Research Program, which is called CDTRP. Actually, it's been funding a lot of transplant research and also doing a lot of helping clinical trials and other things. And we've been fortunate to get some help from them too. Okay, thank you. And also, Dr. West is a former Canada Research Chair, and also she was a past president of World Transplant Society as well as Canadian Society of Transplantation. And also, she is a past chair of the Women in Transplantation International Initiative, and also a fellow of Royal Society of Canada. And the most importantly, actually, she was in 2020. Actually, she was awarded, inducted as Order of Canada, which is it's wonderful. Actually, she has contributed significantly to our Canadian Society for Transplantations area. In addition to that, actually, she is leading a world-leading research group. Actually, she has published almost 300 publications. Among them, actually, the most important may be the ABO-incompatible transplantation, actually, or for pediatrics, and which changed the kind of the way we looked at actually transplantation. And then she currently actually bringing up all of this ABO-incompatibility or ABO-Histocompatibility, actually how we can utilize that or how we can understand that more to do that. And we are very fortunate to have her here, and welcome, Laurie.
Thank you so much. I think it's on. Yeah. Is it on? Great. Thank you so much for that nice introduction and for the invitation to come in person from -16 in Edmonton to this beautiful spring day in Vancouver. Thank you so much for your warm hospitality. So as you've heard, I'm a clinician scientist. I take my clinical role is to take care of pediatric heart transplant patients, and my much larger research role than clinical these days is in transplant immunology, kind of writ large. But I've had a special interest in ABO glycoimmunology for more than 20 years now, and I'll tell you a little bit about that and where it's going. I've just had eye surgery, so I can't tell whether things are better with or without my glasses, so maybe I'll just leave them off, and if I fumble, you can help me. So it's so exciting to come here and talk to you about what we call in my lab ABO-Histocompatibility. And I'm totally aware of the fact that this is kind of a blood audience. And so I want to make a disclaimer straight away that I'm talking about ABO, not in the sense of blood, but in the sense of organ transplantation. And I hope that that perspective becomes clear a little bit, because it's really important to understand how that fits with this concept of ABO-Histocompatibility. So I do have a disclosure. I'm on the scientific advisory board with Avivo Biologics, which I'm sure many of you know, especially Steve and others, is launching some really very exciting work, and I'm happy to be part of it. So let's take off our blood hats and put on our transplant hats. This is organ transplantation. So when I'm referred a patient to work up for transplant, part of that workup, the transplant assessment, involves an immune workup. And generally speaking, we're talking here about HLA, histocompatibility. We're looking for not exactly matching, but towards the least immune risk that we can between the between the recipient of the transplant and the organ donor. And so we start with donor and recipient typing, and that's done by our HLA colleagues. It's done by genotyping, standard practice globally for the donor and the recipient around the world. And we also evaluate HLA antibodies in the recipient to see whether there are any HLA antibodies that will have been caused by some sort of sensitizing event, such as a transfusion or a previous transplant, or in women, a pregnancy. And these could have an obvious impact both on the graft that's transplanted if a donor expresses the antigens to which those antibodies are specific. This could have a detrimental immune effect. We don't actually match, at least not in heart. There's not time to actually match. And so we try and set predictions of risk ahead of time and make either temporizing maneuvers if we need to, or therapeutic maneuvers if we need to, or at least expectant management of what the immunologic risk is going to be. And importantly, we work closely with the HLA team. And also, just to orient you to this, if you're not aware of transplantation, but the clinical decision-making around HLA compatibilities is made on risk assessment and on any interventional strategies available. It's not regulated in any way. That's important. So this is the kind of, in working with our HLA lab colleagues, this is the kind of information we in transplantation have now become reasonably adept at interpreting. This is the profile of HLA antibody analysis using single antigen flow beads. Each bead has a protein antigen attached to it, and the serum of our recipient patient can then be tested on this for a panel of antigens to which they may or may not have antibodies. Again, this is 20th century clinical standard of care for assessment of HLA compatibility. And we can get then an assessment that we call the CPRA, which is something we can hang our hat on and say this patient is highly sensitized or not, and that guides our path forward. ABO is entirely different. And so we tend to think of it in the transplant world. A lot of clinicians think of this as, well, that's the easy part. And so I'm here to convince you that that's not the case. So the ABO system in organ transplantation, as I'm sure all of you know, the A, B, and H, which defines the O histo blood group, these genes encode for glycosyl transferases that add carbohydrate units to form the ABH glycan or sugar antigens. And our tissues and organs that we transplant are decorated with these glycans. So it's not just the blood group system, it's more correctly called the histo blood group system, with histo referring to the tissues. Just for your interest, and probably you know this already, this is not just in humans, but there's phylogeny that shows that these polymorphisms emerged approximately 20 million years ago, and these antigens vary also between species. Back to transplantation then. So my colleagues in clinical transplantation, as well as across many aspects of medicine, have some general assumptions about the importance of ABO antigens and antibodies that are relevant to organ transplantation. Some of these assumptions are, as I said before, that ABO is kind of like HLA, but it's way easier than HLA. Another assumption is that A and B are single antigenic epitopes consisting of terminal trisaccharides to these sugars. It's assumed that the expression of A is probably the same as B without really asking whether it is or not. And it's also assumed that the ABH structures on red cells are the same as those on the organs that we transplant, in other words, hearts and kidneys and so on. So this says all cats have 4 legs, I have 4 legs, therefore I'm a cat. So let's look at this in a little bit more detail because, and we can add to that a couple of other assumptions, and that is that only IgG isotype antibodies are important in transplantation, and yet ABO antibodies maintain an important IgM isotype component as part of what's classically called a T-independent response. And we also assume that erythrocyte agglutination is an appropriate test for characterization of ABO antibodies that are relevant to clinical organ transplantation and to the management there. So let's look at these assumptions in a little more detail. So again, the ABO system in organ transplantation. So unlike HLA sensitization, so-called natural antibodies, and we always have to put little air quotes around that, natural antibodies are spontaneously produced to non-self A and B structures beginning in early infancy, round about sometime 4 to 6 months of age. And these persist throughout life to the antigens that we do not ourselves express. These ABO antibodies can then fix complement and rapidly cause antibody-mediated graft destruction if an ABO incompatible organ is transplanted in the setting of preformed antibodies. And so this is called hyperacute rejection. It kind of looks like this. On the top is a hyperacutely rejected kidney transplant. You can see widespread graft thrombosis. And by hyperacute, we mean within minutes or hours. This really terribly devastating cascade of events happens with complement activation and recruitment of widespread mediators of inflammation and thrombosis and activation of the coagulation cascade. And on the bottom, you can see in an experimental setting, a hyperacutely rejected heart graft where you can see intracardiac myocyte hemorrhage as well as an intravascular occlusive thrombus. So it's not a situation you want to be in. And so in the late 70s, some rules were put into place to prevent accidental ABO incompatible transplantation. But if we go back to our patient that we're working up for a transplant, even before we do any immune assessment for anything else, we know and test their ABO blood groups in case they haven't been determined already. That's the very first thing that happens. And it's phenotype, usually, not genotype. And when we assess our patient's blood group, this typically defines the whole ensuing algorithm of their transplantation, their waiting time. It informs what we might do next because they're stuck in these 4 groups of blood groups. And those don't even go anywhere. Like that's set in stone, that's concrete. Decisions aren't made about that typically anymore unless there are certain circumstances which I'll describe. This is, and then when a donor organ potentially becomes available, we also get the phenotype of the donor organ. And that defines the allocation of that organ. And rules have been put into place, strict regulations typically around how those organs can be allocated to ensure that accidental ABO incompatible transplantation isn't performed. And we have been limited ability to change that, although more so than in the United States, where it's built into a very rigid system that doesn't allow much wiggle room. And again, the contrast is that we have genotyping for HLA and phenotyping for ABO. So what donor and recipient combinations are compatible for solid organ transplant? This is from our article that Jay mentioned that was more than 20 years ago now, looking at ABO incompatible organ transplantation in babies. So if you're in the business of transplanting, you're trying to find a donor for your patient, these are the blood types that are compatible with blood group O. Compatible donors would be blood group O only, and incompatibles would be A, B, and AB, and so on, as you can see. If, on the other hand, you're with an OPO and you're in the business of allocating organs, then your donor's blood type is O. That donor can go to anyone, but an O individual can only receive an O organ if you keep all these compatibility rules in place, similar to blood, of course. And that's all very sensible and has been in place, as I said, for decades. But in contrast to HLA antibodies, again, ABO antibodies are produced spontaneously without specific sensitization. ABO antibodies maintain an important IgM isotype component in addition to IgG, which is in contrast to HLA and other protein antibodies, antibodies to protein antigens, where it's usually a sensitizing event and we're mostly worrying about IgG. In contrast to transfusions, though, and to transfusion medicine, due to the scarcity of donor organs worldwide, we do contemplate and we do undertake intentional ABO incompatible organ transplants. So maybe I'm wrong, but I can't think of too many reasons why one would want to do a transfusion across ABO incompatibility, but this presents itself as an opportunity to expand the donor pool for patients who will otherwise never be transplanted. And in thoracic setting, that means they will die without a transplant, whereas in kidney, they may wait on the waiting list for many, many years without an ABO compatible transplant. And so in certain centers, special programs are developed that allow us to either avoid or deal with these ABO antibodies. Similar to HLA histocompatibility, In order to undertake safe ABO-incompatible transplantation, however, we need to know the antigens that are expressed in the organs we transplant, and we need to know the relevant antibodies or their absence in the recipient, and we need to know their isotype and their specificity. So just like HLA, we need this to really safely manage clinically ABO-incompatible transplants. And again, that's why we come back to the concept of ABO-histocompatibility. It's the same information to make those decisions. So again, this is not just simply A, B, and O. A terminal sugar is N-acetylgalactosamine, and B is D-galactose. But unlike 1H, which is the common impression in the clinical world, at least in transplantation, there are 6 subtypes of each of A, B, and H glycans. So there are 18 ABH glycans, not 3. Each ABH subtype glycan, these are not uniformly expressed across different cells and tissues and the organs we transplant. Erythrocytes differ in their expression of these glycan structures from, again, the tissues and organs that we transplant. And that's not widely appreciated in the transplant world, or really, I don't think in the clinical world much at all. And furthermore, of course, A glycans differ from B glycans. They are not the same thing, but they're often grouped together under the just general heading of ABO. This is what the structures look like. As you can see, these are the chemical structures of subtype 1, 2, 3, 4, 5, and 6. And there are 6 each for H, A, and B, and you can see them laid out here. The first thing that's important to know is for vascular expression, which is really the target of a transplanted organ, mostly, is the vascular endothelium. Those cells express only subtype 2 glycans for both A and B. In contrast, erythrocytes express subtypes 2, 3, and 4 for A, although it's a little simpler, we think, so far for B. So you can see right away that this brings up some problems when we rely on the erythrocyte as a reagent to test for ABO antibodies. So put another way, these are building blocks. These are carbohydrate building blocks like Lego, if you like. And so the erythrocytes have several different ones upon which these are built, 2, 3, and 4, whereas the endothelium expresses only subtype 2. So the first thing we need to know is what's in the heart. Then, if you're a cardiologist, I'm transplanting hearts, what are the ABO structures in the heart? And what we find is that, first of all, only vascular endothelial cells are decorated with ABH glycans, and only subtype 2 glycans are actually found in the heart. So you can see here, I should back up a little bit. The way we were able to determine this is because we generated monoclonal antibodies, a panel of monoclonal antibodies, because you can't really purchase reagents to do this, to do the immunohistochemistry, to define the antigens that are actually expressed in the organs we transplant. And so you can see here that in a blood group A1 heart, this is a pan-A staining, and it's there, and this is a subtype 2 antigen stain, and you can see that A is present, it's absent in B and O, and only subtype 2 glycans, A glycans, are found in the A heart, and conversely for B. In the kidney, it's a little bit more complicated because the cellular structures and components of the kidney are a little more complicated. And in this setting, we see that both endothelium and epithelium express ABH structures, but they're a little bit different. So the endothelial cells, as you can see in green here, in peritubular and glomerular capillaries, you can see subtype 2 glycans, A glycans are expressed there. And also subtype epithelial cells in the distal tubules, as in gray here, also express subtype 2 A glycans. In contrast, subtypes 3 and 4 are only expressed in the epithelial cells and not in the endothelial cells. So as in the heart, the endothelial cells seem to express only subtype 2 glycans. The clinical significance of this, and we've also looked in lung now, where we see a similar pattern to kidney, because of course there are epithelial elements in lung as well, which is a much more complicated tissue. So the nomenclature, of course, is unfortunate because there are also ABO subgroups, and these are not the same thing as the glycan subtypes. These are a different nomenclature. And this is the grouping of the ABO blood groups. So there's not only A blood group, and the most common one is in the A blood group. And most commonly it's ABO-A1, but there are other subgroups, as I'm sure you all know better than I do. Subgroup 2 is the next most common, and then 3, 4, and so on, and also in blood group B. But it's really tricky to make sure that we understand the difference here between the blood groups and the carbohydrate structures. Furthermore, when we look to test our donor and recipient for if they're a blood group A, first of all, most of the time they're not subgrouped. So we don't know, generally speaking, whether they're A1 or non-A1. When they are tested for subgrouping, because we've either specifically asked for it or because there's been an indication, they're usually tested for A versus non-A1, A1 versus non-A1 by lectin binding, by an A1 lectin, and not by genotyping. And so this becomes quite important as well, because A1 subgroup red blood cells likely have A2, this gets confusing, so the subgroup A1 likely has the glycans A2, A3, and A4 on their surface, and we've studied with this as well with monoclonal antibodies and immunohistochemistry, but the proportions are not really clear. And the impact of homozygosity on the glycans that are expressed on the erythrocytes is not well defined. So in other words, if the genotype is A10 versus A1A1, the relative structures of the glycans on the red cell, which may be important, is really not very well defined. And this is true then not only for the erythrocytes, but also for the other tissues. So it gets more complicated than just A, B, and O. On the other hand, A2, these red cells have less A glycans. It's known that the intensity of antigen expression is much less on non-A1 blood groups, on the erythrocytes compared to A1. And they likely only have A2 glycans and possibly weak A4 glycans. And the impact of homozygosity versus heterozygosity is completely unknown. So we're starting to see a more complex picture when we really look for the finer points of these structures than we currently do on a routine basis. And so when we look at trying to cross ABO barriers in order to find organs for patients who will not be transplanted otherwise, we see that we've got ABO identical, ABO compatible, ABO incompatible, but there's also a concept that I'll mention now, and if we have time, I'll come back to, which is called ABO incompatible light. And this is where we take those A2 donors, blood group A2 donors who have less intense expression of the A antigens than A1, and those can be successfully transplanted into blood group O and B recipients who are at greater disadvantage on the waiting list than blood group A individuals are. And so they often linger on the waiting list. This can open a potential doorway for them to be safely transplanted because of the lower expression and the less immunogenicity of this graft in a blood group O, for example, or B recipient. And it's now been shown in this really nice article by Kathy Murphy and Bill Lane that if we go to genotyping from lectins, if we rely on lectins rather for subgrouping, this leads to missed opportunities to do safe A2 into A2 incompatible transplants into O and B recipients. And so we see here the emergence of an understanding that genotyping is really important here in order to take advantage of these opportunities to transplant patients who will otherwise be very hard to transplant. So what's the connection? Of course, genotyping for ABO types the transferase and not the glycan. But then we need to continue to understand the expression on the tissues or the decoration on the tissues of these sugars. And that leads then to an understanding of the ABO antibody profiles of these patients, which reflects, of course, self-tolerance to these structures. But the end result of the transferase is the expression or the decoration on tissues. And if we can mitigate, if there are ways to mitigate the clinical risk to increase transplant access and success, then we'll be able to take advantage of this kind of natural, less immunogenic tissue to get more transplants done. It's one thing to think about transplanting kidney transplant patients who wait many years on dialysis, but in the setting of thoracic transplantation, these are patients who will die if they don't have a transplant. And so it's really very urgent because you list these patients with the hope and understanding that their likelihood of being transplanted may not be very high. And so it's really important to maximize these opportunities by really paying attention to ABO and understanding it with the same finesse that we do HLA. What about the antibodies then? So we can either remove antibodies, preformed antibodies in potential transplant recipients to render their immune situation more favorable to not damage the organ graft, or we can avoid them in some way. And some of the work being done by Steve and colleagues is to remove the antigens so that the antibodies don't have a target. And that's a very exciting thing that we're really happy to be collaborating on. But how do we measure ABO antibodies? So again, this is in your field far more than mine, but I'd like to share with you some new tools that we have for measuring ABO antibodies that I think, especially in the setting of organ transplantation, will be very helpful. So, of course, this is Landsteiner's 1900. This is what? 123 years ago, that the agglutinating substances in serum helped describe the observation of the 4 major blood groups. Well, we kind of do that today, don't we? We do limiting dilutions. We dilute out a serum sample. We use reagent erythrocytes, and the place at which it last agglutinates, we call, that's a titer, 1 in 16. 1 in 16. This is taken by your clinical colleagues as an absolute determination of the quantity of antibody in that serum sample. And that's a mistake. And I'm going to show you a little why and why it would be helpful to have something more accurate and precise. So first of all, there are many variations on the hemagglutination test. And it's remarkable in this day and age that there remain so many variations. And when you just ask your colleagues in different places around the world, well, do you do it at room temperature? No, we do it at 37. Do you do an immediate spin or do you have an incubation time? Is it the gel test or the tube test? Are they commercial cells you use or a local panel of cells? Do you use A1 or A2 cells or and B cells? Who's your vendor? What do they know about how they're stored or whether they're fresh? Distinguishing IgM from IgG isotype can be done using DTT or not, plus an anti-human globulin or not, or both, or neither. And the anti-AHG can vary as well. What is that anti-AHG? Is it anti-IgG, anti-C3d? All of these are variations that are done every day around the world. And then they're reported in the literature, and they don't include any of those details, usually. Usually they don't. And in fact, most of us, speaking as a clinician, don't know any of this stuff. Like, you don't know that your blood bank may be doing these in completely different way to the one across the street or around the world, which makes comparing things very difficult. And the thing is that this is known, and the ABO proficiency testing, surveys, and other studies highlight the variability of detection of ABO antibodies by agglutination. And this is an example of the American College Pathologist proficiency testing using just 4 of those variables, temperature and, that's where I need my glasses. I can't see what that says. Temperature and whether it's tube and with and without AHG. And you can see in these 4 lines here, this is the same serum sample. And look at how different the result will be of an anti-A titer. And so these I'm going to tell you about the work of my last PhD student, Ann Halp, and some of you may know her already. So she's on social media all the time. And she sent out this little informal survey. And she said, calling all ABB people, how do you do? Which do you use to support ABO-incompatible transplantation at your center? Do you use this? And look at the spread she got. And this was just 30 responses, but it's pretty indicative of a situation that's really quite difficult to understand its persistence when everybody knows this is a problem. So, use of the hemagglutination test for risk assessment for ABO antibodies in ABO-incompatible organ transplantation assumes, first of all, that the erythrocyte is an adequate surrogate for the organs we're transplanting. I've already told you that's not the case because they express different structures. Furthermore, there is no specific hemagglutination titer reported in the literature that reliably predicts graft damage. It's all over the place. Moreover, the relative importance of IgM versus IgG remains to be shown conclusively. You can't just ignore one or the other. And by the way, that's true in HLA as well, although we kind of ignore that. Information needed for effective clinical management, including determination of eligibility for transplant, management of the transplant, surveillance after the transplant, as well as after desensitization or removal of antibodies, depends on having the specificities of the antibodies, the quantities of the antibodies, or the reliable determination that they are not there, just as important, to the distinct subtype glycans that are in the graft that we're transplanting. We need to know the IgG and IgM isotype. We also probably need to know the IgG subclasses, which has not also, that's something we're working on in my lab at the moment. It would be helpful to know their complement binding capacity and something about memory. These things are all being done for HLA, and yet we don't do them for ABO. And this is important for effective clinical management. So just a couple of examples, and then I'll show you the new test we're working on. How are we with time? Are we okay with time? I don't know. So my interest in ABO began, as I said, back, well, it was actually Valentine's Day, 1996, when we transplanted the first infant in the world intentionally with an ABO-incompatible heart transplant. He's alive now, a happy young adult with a productive life. He was a blood group O infant, and he received a blood group AB heart intentionally. And that opened the doorway really for infant heart transplantation, because before that, we were losing most of the babies on the waiting list because there weren't any organs. And this was the rationale. ABO antibodies are absent during the first few months of life. This is just a schema of a blood group O patient. They develop anti-B and anti-A antibodies in due course over the first few months. And we reasoned that this would be a safe time to introduce an a graft expressing non-self ABO antigens safely. And we were right. And it was actually as simple as that. We had to be very careful of the blood products that were being administered. So we couldn't prime the pump with blood group O plasma that had anti-A and anti-B antibodies, or we'd be defeating the purpose. But this natural window was there, and it had been known for years that babies don't make these antibodies. But the rules were so rigorous and so strict that nobody had really questioned them before. So this gave us so we reported the first 10 patients who safely received ABO-incompatible transplants. Most of them didn't need anything to be done ahead of time. We just needed to avoid giving them antibodies that would damage the graft. But one question that arises then, well, okay, as they grow, they start to make these antibodies. When does that window close? What are those antibodies exactly? What's their isotype? What's their specificity? Are they all the same? We don't know the characteristics of this, so it's very hard to figure out, well, if we just stop at 6 months or 8 months and say, well, that's an arbitrary place to be so-called safe, and when 100% of these babies are going to die without a transplant, that concept of safe takes on some nuance. But anyway, we don't have, hemagglutination is not going to tell us the answers to what those antibodies are. What was really exciting, so this became adopted, many centers around the world use this protocol now. What happened as the children grew up was equally exciting from a scientific perspective, and that was that immune tolerance developed in these babies, similar to the work that had been done by Peter Medawar in the 1950s on neonatal tolerance. When these antigens were introduced into a young immune system, these babies went on to develop anti-B antibodies, so it's got the natural internal control that's lovely about this setting. Blood group O individuals went on to develop anti-B naturally, normally, but they never developed anti-A. And that was very exciting because it was the first time neonatal tolerance had really been reported in the human setting by similar mechanisms, which would have been deletion of antigen-specific B cells, as had been described in animal models. But then you say, okay, so every so often a baby comes along who's got a titer of say 1 in 16, and everybody panics and says, is this dangerous to the graft? But how are we measuring it? We're doing a hemagglutination test to measure it using erythrocytes that express antigens that are not expressed in the graft. And so it became clear after the first, I mean, there aren't many of these, this is not a big field, but it became clear that these antibodies were not actually graft-directed antibodies in the few cases that they developed. And that was helpful. And it was really the fine specificity of tolerance that was so exciting. So in other words, blood group A, subtype 2 glycans are the only thing in that heart graft. But if you're measuring the antibodies with erythrocytes, you're getting anti-A antibodies to subtypes 3 and 4 glycans. And so we showed that the tolerance with the fine specificity of tolerance in this setting, which is really a very interesting thing to see. It was only subtype 2 directed antibodies that they lacked because that's what they were exposed to. Okay, what about kidney? Well, so kidneys are by far the biggest number. You have more leeway. You have the possibility of living donation. You can plan things out. It's not the middle of the night where your opportunities to intervene and remove antibodies are obviously much more limited. But kidneys can be performed not only from deceased donors, but from living donors as well. And in fact, ABO-incompatible transplantation has been performed and is, I would say, increasing. There are specialized centers that do it. They take on the immune risk. They say, we are ready with plasmapheresis, with desensitizing, with taking on these immune risks. And they report that the 1-year uncensored graft survival in ABO-incompatible recipients is really very good. It's comparable to ABO-compatible. But the payoff is that almost half of reported causes of death in ABO-incompatible kidney recipients were due to infection versus 13% in compatible patients. That's a big difference. And that, we think, reflects a tendency towards over-immunosuppression in what's presumed to be a high immunologic risk without knowing whether it really is or not. And that reflects uncertainty in risk assessment and inadequate precision in risk assessment that then is not there to guide clinicians into whether this is truly high risk or not. So the major limitations, as I've told you, to ABO-incompatible transplant literature are that there's no standardization in what we're comparing in different centers. The antibody isotypes, the way of detecting them varies from center to center and varies by method, and this is especially relevant after plasmapheresis, and the subtype specificities are not tested. So how can we improve this? Taking you back to what we expect is standard of care for HLA antibodies. So think about it. For the heart, could the risk of an ABO-incompatible transplant from an HLA favorable donor offset the high risk of dying while waiting for an ABO-compatible donor? That's what's being done now. These highly sensitized patients are waiting for an ABO-compatible donor. The clinical programs don't even think about the possibility of crossing the ABO barrier in heart. For kidney, if we had an ABO-incompatible HLA identical living donor, if you're a nephrologist in the room, raise your hand if you think that's a lower immunologic risk than taking an ABO, waiting for an ABO-compatible donor who's also HLA compatible. Most nephrologists we talk to say that would be preferable. That would be the lower risk if we could understand the antibodies. So this is our view, and I'd like to introduce Ann Halpin here because she really came up with this concept. HLA antibody testing has evolved over the last 20 years. Single antigen flow beads have revolutionized how we manage immune risk in HLA, but it's taken many steps. These are the steps, where we went from donor cells to ELISA to flow cytometry, and now single antigen flow beads. We kind of think of this as ABO testing still being back here in those early stages, so that we're still using red cell agglutination with or without some enhancements that may help a little bit. But do we really need to go through this whole evolution? How about we think ahead and take the principles that we've learned from HLA antibody detection and the tools that have been so important in the evolution of that field and work on that for ABO. And that's exactly what Ann did. for her PhD studies. What can we do to improve antibody detection? Develop a Luminex single bead antigen assay for reliable and accurate characterization of ABO antibodies. So the components of this assay are very similar to those used in the HLA lab. We took ABH antigens, all subtypes that were synthesized for us by our wonderful colleagues in carbohydrate chemistry at the University of Alberta. And we coupled these to individual Luminex beads. We included a no antigen negative control bead, the way HLA lab does. This is done like by a cookbook, a way the HLA lab does it. But you can see that right away, it includes a lot of things that we just don't have in agglutination, cannot be done in agglutination testing. We coupled alpha-gal as a positive control bead. We included negative and positive control sera, and a tiny amount, only 20 microliters of serum is required to run this. And the other piece is that this can be run in an HLA lab who already have all the equipment to do this because they do it every day around the world. So again, the blood group antigens were synthesized in a long-standing collaboration with Todd Lowry and Chris Cairo and others in the University of Alberta Chemistry. And this is what the assay looks like. Again, it's fairly straightforward. I don't know how whether all of you know about Luminex testing and how it works, but the beads all have different intensities of color. And so you can fix a structure to each bead and then be able to detect it by its color intensity. And that way you can tell what the structure on the bead is. So these are single antigens fixed to single beads of single colors. And then a secondary antibody is added to detect either IgM or IgG. And that secondary antibody is further tagged so that you can resolve this out. And it's put into a Luminex machine that every HLA lab in the world uses, as I said, every day. And you get a readout that's very similar to that panel I showed you of HLA antibodies. You have IgM and IgG isotypes to all the different targets that you're looking for. This is an example of what it looks like. This is an ABO-A individual who, as you can see, has some antibody to the trisaccharide, but not to any of the glycans that are expressed here, because those would be self-antibodies, but has anti-B, both IgM and obviously IgG, to the B structures. And I'll show you just a couple of more. This is a blood group O individual who has, and it's very interesting, look at all the IgG, lots of IgG. So it's clearly not just IgM, which you can see in the gray here. And the vascular targets of these antibodies, if you were to transplant an A organ in, would be seen here, the A2, which is not as high as those that would be picked up with an erythrocyte agglutination assay, which is 2, 3, and 4. So this might give you a high titer, but relatively, positive, but not sky high, actual quantities of antibodies. And the B as well, you can see that the B IgM quantity by mean, I should tell you, this is mean intensity fluorescence here on the y-axis and the different glycans here on the x-axis. This is a blood group B individual. And again, you can see that there's a lot of IgG detected here. But here, the anti-A profile in this B individual to the vascular target is really low for IgG and as well for the B2 target. Well, of course, that's self. This is a B individual. So we've worked on this now for the last couple of years. The A and B antigens are coupled to all beads with antigen density that's very similar. And we did a proficiency testing kind of exercise with HLA labs around North America and in Europe to see how these beads performed in the hands of people who'd never seen them before, but do HLA antibody testing every day. And they actually did very well. There was good concordance between all laboratories, and there was good intra-laboratory reproducibility. So then we start to get into new data that's really interesting because it's not always expected. So here's looking at blood from ABO individuals, and you can see from the IgG on top, IgM on the bottom, anti-A in blue and anti-B in orange, you can see from this panel here that in blood group O individuals, I hope that's big enough. Blood group O individuals have a lot more IgG anti-A antibody than blood group B individuals. That's really helpful and could be very important. And yet the IgM is not so variable. So this quantity, this much higher quantity of IgG may be very interesting. So the question then comes up with, well, how do I compare it to what I think I know with a hemagglutination assay? And this is where things get really interesting. So this on the top is anti-A. This is IgM. MFIs here. The MFIs again are on the y-axis. That's mean fluorescence intensity. And these are the hemagglutination titers here across the x-axis. This is IgM on the left, and we add in IgG on the right. Well, what you can see here is that at each hemagglutination titer step, at each titer step, so for example, this one is 1 in 128, there's a range of antibodies, all with the same titer. There's a range from almost none to really quite high. When you add in the IgG, again, at the same titer, it's the same titer, you can see that high levels of of IgG are detected here. And again, these are all at the same titer. I might have hung my hat on that titer and said, this is either too high or too low. Down here, at a negative titer, there's still antibodies that can be detected, even though the titer is negative. And the anti-B has a similar profile. So we know that IgM typically drives hemagglutination, but IgG also participates. And what we can really see here is that we're missing the detection of a lot of antibodies because of this overlap in the quantities of antibodies that are actually at each titer step. Every titer includes a range of IgG and IgM antibodies. And so really, when we make a clinical decision on that titer, we really know nothing because it doesn't give us anything we need. So what are we missing? Here's a case in point. We were contacted by a colleague in Europe who said, I have a kidney transplant patient, a blood group O individual who had a B transplant. His anti-B antibody using an AHG enhanced, using no AHG to enhance the titer was 1 in 2. With AHG, it was 1 in 8. We thought we were fine. We went into the operating room and it was a catastrophe. And there were no other antibodies that we could blame it on. We couldn't blame it on HLA antibodies. Do you think you could test this serum and tell us if there's something in it that we missed by agglutination? And here's what we found. High levels of IgG antibodies, specifically to the vascular target here, the B2 glycan in the kidney. And this was not reflected in anything that was concerning about the titer. So this is an example, I think, this is one case I'm just sharing with you, but it tells us that unpredictable nature that bothers surgeons in going into the operating room with an ABO-incompatible transplant, may be related to something like this, where this is unexpected based on currently used technology. And maybe it was a discrepancy between the kind of AHG that was used, or maybe it was subclasses that we don't understand yet, but this could be very important. So the take-home messages are that the agglutination test does not provide subtype-specific antibody determination that we need to assess these risks. Each red cell titer contains a range of IgM and IgG antibodies, and the titers will vary depending on the agglutination methods performed. So we think that improved assays could be used to really improve the risk assessment. And that as we move from this is a kind of a table that tells you the pros and cons of hemagglutination of our previous iteration of solid phase technology, which was a glycan microarray, and of the current bead-based assay, which is that it's we can determine the specificities, we can readily distinguish the isotypes. The readout is documented. In a hemagglutination assay, it's not documented. It's read and then it's gone. You can't even go back and look at it again. So it's very reproducible. The laboratories have this equipment and use it every day. The expertise is there in the HLA labs. And really it's best suited, we think, for measurement of ABO antibodies in this setting. If I have just a minute, I'll share just a little bit about the A2. We don't do that many A2 to O or A2 to B kidney transplants in Canada, but it's probably more common than what we'd call a full ABO, and it offers more donor options. And you don't have to actually remove any antibodies because usually they're very low, although we don't know that, and sometimes mistakes are made, or they're not mistakes, they're just limitations of the current technology. And so there's also not a consensus on how to test for antibodies with even the hemagglutination assay. But one of the programs that does the most of A2 to O or to B is the Midwest Transplant Program. These sera are DTT treated to denature the IgM, and they don't add IHG to enhance IgG, so that it will detect more the high levels of IgG. And what we found in 2 different patients tested using their protocol for agglutination and adding those to our beads is that in these two individuals had identical anti-A titers of 1 and 4, and both were transplanted successfully. But the difference that we looked at in trying to understand what was there was that depending on whether you used AHG to kind of enhance the IgG or use DTT to get rid of the IgM, or rather didn't use DTT or AHG, kind of reflecting an IgM sort of flavor, is that the titers of 1 and 4 changed dramatically to 1 and 256, which would have suggested that this was not a safe transplant to do. And yet, when we tested it by Luminex, you can see that, again, these are A2, that the quantity of antibody here reflected by this MFI against the A2 glycan is really not in a high range, whereas the IgG components are much higher in this case in particular. So we think that this will be helpful, and what may also be helpful is using A2 erythrocytes to do the hemagglutination titers here. So just a moment on what's next. We still have to understand these differences between this new technology and what the clinical world tends to understand, and how to best understand that new information. And that requires standardization as best we can of the hemagglutination titer assay that's being done in order to compare them. We have to look at the pathology in these biopsies, endothelial and epithelial components, and probably include molecular methods to define the signature of ABO-incompatible transplants. And again, it may be helpful to use A2 erythrocytes in helping us understand how best to deal with this new information. This is the kind of thing that we're hoping we can get by sending to we have a lot of colleagues now out there asking for us to test serum or to try and get these beads out there. And what we need to do is make worksheets, we think, where we say, how is this done at your center? This is the DTT method that's used by the Midwest program, where you use DTT, you've got many vendors for the cells, and you're using A1 cells. But we need information from all the other centers then that show maybe actually this is how your titer is measured at your center. Maybe it's measured with AHG at 37 degrees in a tube test, and you've got vendor Y, and maybe you're using these cells and so on. But once we can get a handle on this information by including all these variations, we think it will be easier to move forward. There are many studies that now are underway looking at retrospectively at kidney transplants, prospectively at lung transplants, memory, and all these other things. And I think I'll just leave it there. And we need your help in doing these tests. And I'll just acknowledge that the people who worked on this in my lab include the people you see here. Again, this was driven by Ann Halpin, who is our HLA lab director in training, but has also now ideally placed to look at ABO-Histocompatibility. And I'd like to also just acknowledge the chemists who make these wonderful toys for us. It's astonishing to me that these things can be made and used the way they can, and it's been a really enriching, longstanding collaboration now with our lab and with their predecessors at ChemBiomed at the University of Alberta. And finally, to all of our many collaborators working with us on these, including Jay and Steve and others. And so I'd like to thank you very much again for the invitation, and I'd be happy to answer any questions if you have any.
Great, thank you very much. So questions from the audience. So please switch on the microphone.
Hello, that was amazing. It's so complex, and I'm a hematologist. I just have a couple of questions. One, I'm curious, do you have any idea why the glycans are differentially expressed in different cells, different tissues? Does anybody look at the function of these other than to cause great angst in terms of transplantation?
I don't know. It's an interesting literature because it's been kind of explored for a long time, and yet the tools have never been ideally positioned to even look at their presence in different tissues, much less their functions. I mean, we know from some work that we've been doing with the Association of Blood Groups and COVID that they can, in some cases, act as co-receptors for viruses, and perhaps that's part of the reason. But I don't have another answer yet, but some of these new tools that we have, we hope we can look into that and look at, well, okay, these are potential reasons why this might have a mechanism here into some specific function.
The second question is, the patient who received the young child, the baby who received the ABO-incompatible heart and developed tolerance. So I don't know how long that tolerance lasted. Was that life? It was at the moment of its lifelong. Is that a strategy for vaccination?
Yeah, well, I've thought a lot about that. So these children as a group are tolerant. And as long as we've looked at them, like Caleb is still in touch with me. Every so often we look at his blood. He still has no anti-A or anti-B antibodies, blood group O individual. And it works. Of course, it's more interesting when it's an O individual who receives a B or an A, because then you can look at their third party antibody. As so far as we've tested, they're there. I mean, we know the organ is unlikely not to be expressing those antigens because the glycosyl transferases are still being produced. So as long as those antigens are present, they seem to remain tolerant. What was the, as a vaccination? Yeah, I know.
I mean, a lot of us are going to receive transfusions over our lifetime.
Exactly. Wouldn't that be an interesting way to go? I think I woke up in the middle of the night one night and thought, oh, but to my knowledge, it hasn't quite been brought to fruition. Thank you.
It's not. Oh, there it is. I don't have a blood background. You mentioned A1 versus A2. If these hemagglutination assays are so bad, how do you know if someone is A2 or A1?
This is a great question. Thank you. So in the clinical world, we just ignore it. And so you think you're doing an ABO identical transplant. We were talking about this, and maybe Steve and I were. You think it's A to A. But you don't know that because most of the time you don't even have a subgrouping by lectin. You don't have a subgrouping at all. So you think it's A to A. It could be A1 to A2, which would be immunologically perhaps a bit risky. It could be A2 to A1, which probably wouldn't matter. Or it could be A2 to A2, which wouldn't matter. But we don't know because we don't routinely do subgrouping, and we certainly don't do genotyping. And that's why the whole thing has to come together. This idea of histocompatibility, you need not only the antibodies and the knowledge about them, but the antigen structures as well. You need to be doing genotyping of your patient at the time of assessment, just like you do for HLA. And you need to be doing genotyping of your donors, just like you do for HLA. And again, this is all done every day in the HLA lab, the genotyping and the Luminex assays. So the infrastructure is there to do it. And then we would not only learn maybe some of those non-HLA antibodies that cause damage, maybe those are actually a setup like that, where it's not, in fact, ABO identical. It could be that there's a difference there between the groups, the subgroups that we don't know about.
Yeah, those are very nice and very, I feel quite shocked at a couple of levels. So first one is just that it staggers me that the tests are so different across these different centers. And I think you maybe gave the reason at the end that there's vendors specific, and maybe they're carving out different IP space in order to find a niche. But that's a philosophic question. But my question is very similar to Ed's. And so I should have been aware of this, but I wasn't. So you're saying then that on each cell type, like erythrocyte, that every glyco on the cell surface is of a particular branch morphology to give it that A or B or AB or a different subtype. So every N-link glyco on every protein that's being expressed on the cell surface is all the same. Because otherwise you'd be getting B and O's and all those other ones coming up on other proteins.
Well, all I can say is that the tools that we've used to look at the glycan expression on the tissues that we have had access to, using the monoclonal antibodies that we developed, when we looked at 20 native hearts, there was no difference in the expression between individuals. And the only thing in hearts that we saw was subtype 2A glycans and subtype B A glycans. I don't know if that's exactly answering your question, that every glycan has the...
Well, otherwise, I mean, you'd have, if you've got different N groups, N capping groups on the different N-link chains, then you've got potential for having different antibodies, and some of them would be cross-reacted to the different blood groups. And so, I mean, with glycomics, we're doing, I mean, probably Steve knows the answer to this, but that's the implication, that every group is kind of the same. And therefore, I guess you can have tissue specificity. The endothelium versus the epithelium in the kidney will have a different glycosol transferase pattern or some coding there that enables different types to come up either on that cell type versus that cell type, and that would reflect function, circling back to the functional question.
Yeah. Well, I don't know how much that would impact the antibodies that come up, though, because although those are...
Well, self-tolerance, right? So at self-tolerance
exactly.
stage, everything's going to be sorted out in the thymus, and I guess now through macrophages taking in antigens from grafts and things in now, somehow to get tolerance in the early days.
Yeah, I mean, it's self-tolerance, but the stimulus for the production of the antibodies in the first place, which is always said to be a cross-reaction to non-pathologic gut flora, well, that's not true either. Because if you raise mice in a germ-free environment, they still produce anti-A and anti-B antibodies. So there's a lot about it that still, I think, is incompletely understood. And until we have adequate tools to ask those questions and to explore those things, some of that dogma is going to remain unchallenged.
Dogma's never has a chance of surviving. I think if it's more than 30 years old. Yeah.
It's way more than 30 years old.
Yeah, that's got no chance of surviving. Yeah.
I mean, if Landsteiner's method is still the one we use now, that's kind of frightening in itself, isn't it?
So I just wanted to, you asked, is it tolerance or is it immune suppression that you don't get antibodies? And I think you mentioned the third party. You can measure if it's an A and the O, you can measure anti-B. And what happens then if you didn't?
Well, my point is that it's,
I mean, you couldn't get a better control.
And it's the perfect control. You know, isn't that a wonderful setup?
Yeah.
Well, we think it's deletional tolerance,
Yeah, so
because
it's
when
true
we look for
tolerance.
antigen-specific B cells
Yeah, true
in
tolerance.
those individuals, we don't see them. And so, and we reported that a number of years ago, that those cells actually seem to be deleted from the
Sure.
from the B cell repertoire. So we think it's not suppression.
So this is kind of related to the first and second last question. So if you genotype, say, a red blood cell for glycosyl transferases, can you predict what kind of glycans there might be in the heart or the kidney based on what is in the genome?
I don't know the answer to that. I mean, I think there's an awful lot, at least from that I don't know about the glycosyl transferases. You know, clearly all those little changes in the structures of the glycans must reflect some functional difference in the glycosyl transferases, but that's the extent of what I know about them. And again, I think as we have more refined tools that allow us to ask questions like this, maybe that, I mean, there's a whole slew of questions that haven't been addressed that maybe now could start to be. It's a great question, though.
I mean, the problem with glycans is they're not, of course, directly encoded in the genome, right, themselves. So And it's going to be dependent not only on the identity of the enzyme that puts that on there, but its localization and the availability of the donor sugar. It's a complex beast. So, but who knows? Yeah.
So when we go back to saying A, it's just A, B, and O, right? So simple.
Can I ask a question remotely?
We are coming to the Zoom.
Yes. Hi, I'm Tyler Smith, a hematopathologist. I hope you can hear me okay. It's excellent talk, and I think it really highlights the challenges sometimes when you have an area that's kind of an unexplored overlap between HLA transplant world and hematopathology blood banker world. It seems, and I think really you've highlighted that the largest issue is assay standardization and that kind of lab clinical correlation. Have you worked with Robert Livsky? Are you aware of Robert Livsky? He's a hematopathologist, but he's also HLA director in Dalhousie. And
He's a
also,
collaborator, yes.
good,
And
yeah.
you're absolutely right. It's people who can kind of sit on that, and Ann Halpin is the same. And we're kind of discovering and we hope an expanding group of individuals who can join ABO and HLA in that way. But that's a really important thing to point out. Thank you.
Okay, thank you.
So you mentioned that the Luminex assay could give us a much more precise measure of the antibodies, but I'm just wondering, so like what would be the next step of actually determining like what antibody threshold would be okay to use, or like would have like a lower risk? Would you would you do that, like would you have to do that in actual surgery and trial and error basically, or is there like another model that you could use before using that assay in the actual transplant?
Well, there's a lot because our HLA colleagues are self-described as hoarders. They save every serum they ever get. And so so we can do a lot retrospectively, we think. We can look at the serum that they have from transplants past, from patients who were assessed for transplant but declared ineligible. We can retest their serum with this and compare it to hemagglutination. I think we really need much more understanding of those results before we can say in my own clinic, for example, I think that's a safe ABO transplant, and what you're getting as a titer is not accurate because I can measure the antibodies and they're not there to the glycan of importance. But I think we really have to really understand a bit more, and so that's going to require us continuing to test this until we have, until we can look at conclusions and then correlate them with clinical outcomes. I think there's someone in the back there.
The ABO antigen. Do you think after transplantation they can raise back?
So if the antigens are removed, will they come back?
If you want to remove, maybe you are efficient and you remove this antigen before a transplantation. Do you think it's possible over time they will grow back?
So this is work that Steve is that we're working on, but it's Steve's product, Steve Withers here, who developed these enzymes that remove the antigens. It's almost certain they will come back.
Okay.
The kinetics of how quickly they come back and whether they're altered in any way is something we're currently studying. We have a transgenic mouse model that helps us look at. It's kind of hard to study because mice don't have these structures, but with the transgenic model we can get some information. But it's almost certain that they will come back, and that's been studied a little bit also by Yuri Galili and his work a decade or so ago with alpha-gal as another carbohydrate structure.
Thank you.
Thank you.
There's no more. Thank Laurie for
Thank you.